Hello and welcome to ASO demola, and today's lesson we are doing meiosis and fertilization. We're going to cover zygotes, gametes, crossing over, independent assortment, the whole lot. Let's get straight into it.
Okay, so we're going to start this topic by talking about gametes. Now, your gametes are your sex cells, and they contain half the number of chromosomes, which we're going to denote as N, or we can say it's haploid. Yeah, your normal cells are diploid, so they contain 2N chromosomes. In a human, N is 23. Cool. But yeah, gametes are your sex cells, and they can include your sperm and your egg cells. So we're going to talk about both, and then we'll link it into its function in fertilization. So a sperm cell, let's have a look. You can see this is a diagram of a sperm cell, nicely drawn by yours truly. But as we can see here, in the sperm cell, we've got at the right hand side the flagellum. Yeah, that's the tau. We're going to talk about definition in a second. We've got this bit in the middle, which we haven't actually labeled yet. We can see that it has a lot of mitochondria. But if I highlight this is your mid piece. Yeah, so the mid piece of your sperm cell contains loads of mitochondria. Yeah, then you have the head. Now, one thing important about the head is its shape, so it's streamlined. So that will help it to move through liquid as it's going towards the egg cell. What you can also see in here, it's got its haploid nucleus containing half the number of chromosomes. Cool. We're going to talk about why it has half the number of chromosomes in a bit. And you've got an acrosome. So we're going to talk about definitions now, but you've seen the sperm cell there. Cool. So some of the things we need to define an acrosome, yeah? It's in some ways a type of lysosome. So it contains digestive enzymes. We're going to talk about its function again in the next bit. Flagellum, yeah, that's tail-like structure, propels itself through fluids. If you've seen the video about cell structure, you would have known about flagellum. Mitochondria is another one you would have known. So aerobic respiration and ATP synthesis. So that's a bit about the sperm cell. Now let's look at the egg cell. So, of course, we're dealing with something a bit different here. But again, we have a lot of similar tones. So we have this outer bit. Yeah, it's like this jelly-like glycoprotein layer called the zona pellucida. Inside of the cell now, so you've got the zona pellucida surrounding the cell membrane. Inside of the cell, we've got lipid droplets. We're going to use those for energy. We can use lipids as a source of energy if the glucose gets low. We've also got a mitochondria, again, aerobic respiration. Cortical granules, we're going to talk about in a second. And a haploid nucleus, again, half the number of chromosomes. So, zona pellucida is a jelly-like protective layer surrounds the cell. Yeah, we're going to talk about it again in the next bit. And the cortical granules, again, we'll talk about, but they secrete chemicals to harden the zona pellucida. And we'll talk about why the zona pellucida needs to be hardened next. Okay, so now let's talk about the actual process of fertilization. And we're going to break it down into 3 steps. So number 1 is the acrosome reaction. Now, what's going on? So you have that sperm cell, yeah? After ejaculation, it's going to start moving towards the egg cell, moving towards the uterus, yeah? Now, when it reaches the uterus, finds an egg cell, it gets to the zona pellucida. And like we said, that's a protective layer, right? So the sperm cell shouldn't have an easy time getting through it. Now, that acrosome, right, if we zoom in, is going to fuse to the cell surface membrane of the sperm cell. So you can see it's getting closer to the cell surface membrane. When it does that, we're actually going to release these digestive enzymes by exocytosis. And they're going to do what? Digest, right? So they're going to digest that glycoprotein layer, the zona pellucida. They're going to digest some of it so that we can get a pathway for the sperm to get to the egg cell. So you can see this sperm is a man on a mission. It's going towards that egg cell. It's going towards the egg cell membrane. Yeah? Cool. And eventually, fingers crossed, you would hope so, it's going to get there. And that's what we're going to talk about in the cortical reaction. So your sperm cell has got to the egg cell membrane. It's fused with it. It's going to now allow for your nucleus here to skedaddle into the egg cell. Yeah? Cool. The cortical granules now, when they realize that the sperm cell membrane has met the egg cell membrane, they're going to go towards the cell membrane of the egg cell as well. And they'll fuse with it and release their chemicals into the zona pellucida by exocytosis. Yeah? So you can see that written here. Now, those chemicals, when they're in the zona pellucida, they're going to harden it. So once it's hardened, those digestive enzymes that used to allow your sperm cells to make a pathway to get to the egg cell, they can no longer digest it. Can no longer digest it because it's so hard. And that's going to prevent a concept that we call polyspermy, which is when the same egg cell gets fertilized by multiple sperm. Cool. Now what's going to happen is we've had the fusion of the membranes. The haploid nuclei of the sperm cell and the haploid nuclei of the egg cell are both in the same place, right? And they're going to fuse. So when they fuse, we form what we call a diploid zygote cell. So zygote cell, just good to remember, diploid because we had 2 haploids, so N and N joined to make 2N, which is your diploid number, yeah? And then that zygote is going to continue to divide and divide by mitosis, eventually become an embryo, the whole shebang, and then you get a human, yeah? Hope that made sense in terms of the stages of fertilization. Okay, now we talked about gametes, right? We talked about what they do in terms of fertilization, acrosome reaction, cortical reaction, fusion of nuclei. But how are these made? Now, meiosis is that process. So meiosis is the process by which we're going to make our gametes, right? They're gonna occur in your sex organs. So, for example, your ovaries, your testes, yeah, that's where meiosis is happening, and they're only happening to germline cells, right? So your somatic cells do mitosis, germline cells do meiosis. Now, meiosis is very similar to mitosis, right? Make sure that we know the difference: meiosis versus mitosis. Now, meiosis happens over 2 rounds of division. Yeah, meiosis one, meiosis two, nicely named. These stages are exactly the same as mitosis. So you have the first meiotic division, where you have prophase one, metaphase one, anaphase one, telophase one, and you have the second meiotic division where you have the same thing as in prophase two, metaphase two, etc. You produce 4 haploid gametes. So 4 haploid gametes. Yeah, that's what you produce. And you also get genetic variation. So these gametes are not genetically identical to each other. They're not genetically identical to the parent cell as well. And that's because they now have a different combination of DNA or different combination of alleles. Yeah? But how? How's that happen? So there's 2 main processes by which it happens actually. First one is crossing over. So define it: exchange of alleles between non-sister chromatids, and we've talked about sister chromatids before, right? In the mitosis video, which you can watch on the channel. Now, crossing over happens in prophase one. So we know prophase, your nuclear envelope dissolves, chromosomes decondense, etc. That's when this is happening. And when you have non-sister chromatids that belong to the same chromosome, you have what you call a homologous pair. Yeah, so you have a homologous pair. You have, say, purple is the maternal, green is the paternal. Yeah, but you've got those two. Now, what they're gonna do is they're gonna join together in some way. So you can see here, if I circle it, at this point here, we have the joining of the non-sister chromatids in the homologous pair, and that point is what we call chiasmata. So they're joined at the chiasmata, and then what's gonna happen is a bivalent is gonna form. So this here is called a bivalent. Yeah. Now, parts of the non-sister chromatids are gonna break off, right? They break off and they swap positions. So you can see here that the green has come onto the purple, and here the purple is coming into the green. Yeah. Get rid of those now. So what's happened now is you form what we call recombinant chromosomes, and you can see here, because we've exchanged parts of the alleles on the non-sister chromatids, that is why we can have recombinant chromosomes creating genetic diversity. Yeah, just know that it can also happen in multiple places. So, like for example, say the top's linked as well, you don't have crossing over at the top, so we can have a lot of different combinations forming crossing over. The next thing we do is independent assortment. So unfortunately, don't have a nice diagram for this one, but essentially it's the random arrangement of the homologous pairs during metaphase one. So you know in metaphase you have the lining up of your homologous pairs this time along the equator of the cell. In metaphase two, it's going to be the sister chromatids, just like we had with meiosis. But in metaphase one, it's the homologous pairs. Yeah, they line up along the equator of the cell, but the way that they do that is quite unique. So maybe one time you're going to have maternal on the left, paternal on the right, or the other time you have paternal on the left, maternal on the right, different combinations, right? That is going to help us to make a lot of different unique ones, which is going to mean that we get genetic variation because you don't know which combination of maternal, paternal chromosomes are going to end up in which cell. Yeah? To get a total number of combinations, we do the formula just 2n squared. Yeah, so you've seen it right now, 2n squared. N being 23 in a human, you get 2,116 different combinations. Yeah? Hope that makes sense. Okay, this is quite content heavy, but we do have our questions, so you can see all 3 of them there. First one, you're discussing about stages after the acrosome reaction. Then you're talking about why this sperm cell might reduce fertility, and then we're talking about crossing over. So, pause the video, have a go, and then we'll get into all the answers. Okay, let's run through some explanations. So, first one says we're talking about stages after the acrosome reaction. So we know that's when we're creating a pathway for the sperm to get to the egg cell. Well, hopefully, eventually, the sperm cell is actually going to then fuse with the egg cell. So that's the first thing we want to talk about. Yeah. Then we need to kind of talk about the cortical reaction. So you know about your cortical granules. They fuse with the cell surface membrane of the egg cell. They're going to do exocytosis of the cortical granules, and then that's going to that gets released into the zona pellucida, jelly layer here, and it hardens it and thickens it. Yeah, that's kind of what they want to talk about in terms of what happens after the acrosome reaction. Your key is to say the cortical reaction. Yeah, cool. Then you could see on that diagram the person had teratozospermia. Don't know if I pronounced it right, but you could see they had two flagella. Yeah, so it's already pointed to that being something that's bad, right? Eventually, at the end of the day, we should be knowing that the main thing that we are preventing is fertilization. That's probably where I would start. We can't do fertilization because the sperm isn't going to reach the egg. At the end of the day, that's really going to be the main thing. Then we can just think flagella, they're involved in movement. So probably having two of them means that the sperm can't do it as well. So maybe I'd work backwards from the answer. You know what I mean? But yeah, those points should be fairly decent to get. And in terms of this one, it comes to the understanding of what a homologous pair of chromosomes is, right? So if my chromosomes are homologous, that means that they have all the same genes and the same gene locus points. Yeah, all the same genes and the same gene locus points. The only differences might be they might have different alleles, right? But if we don't have homologous chromosomes, right, we can't have crossing over. Yeah, there are some chromosomes or there are some sections, some genes on the X chromosome that aren't on the Y and vice versa. Hence, this crossing over can't happen if that's not present. So yeah, we're literally going to say can't really happen between the X and the Y chromosomes because they're not homologous. That would be what I'd go for. I wouldn't even really say it can happen in some parts. I'd just say not homologous pairs. Doesn't happen. Okay, and finally we're at the A-star tips. So stages of fertilization, very important in terms of acrosome, cortical, and then the fusion of the nuclei. Adaptations of the gametes as well. Why they're beneficial. I will always be such a strong advocate for linker feature to a function. Yeah, feature to function doesn't miss in biology, doesn't miss. So yeah, make sure that you can explain why is there an acrosome, why is there a haplonucleus, why is there mitochondria, you know what I mean? And then comparing and contrasting mitosis and meiosis, we haven't really focused too much on it in this lesson, but it's a question that comes up really commonly, so just be really happy. You've got that table down, you can pause, have a look. But those are going to be the main differences between mitosis and meiosis. Yeah? Stages of meiosis is something that you don't need to know. In the SNAB specification, you definitely don't. I think most of them aren't going to ask you in depth about the stages of meiosis, but at the end of the day, if they do, it's the same as mitosis. Yeah. So your description for anaphase or telophase is going to be exactly the same as it would be for mitosis. Maybe just be aware that if you're describing metaphase 1, you need to say homologous pairs instead of sister chromatids. And then neglecting the acrosome and cortical reactions. I think it's something that's just often forgotten, to be honest. Just don't let that be you. Yeah, so just make sure that you're aware of it, you're happy with it. They aren't that complicated to learn, to be honest. So, hope that video was helpful. Any questions at all, drop them in the comments below. I'll do my best to answer. If you enjoyed the video, if you found it useful, please like, subscribe to the channel. We've also got our TikTok and our Instagram, both at A Star Demola. But that's all for me. I'll see you in the next one. Have a good one.